It is completed, it is activated protein, and it's completely different than just basic extracellular vesicle. MSCs from umbilical cord lining tissue are different. They specifically mediate tissue protection and regeneration. They are much more broad spectrum and broad reaching than a typical cell. Many cells are very specific. These have a broad ranging category, and they're not specific to a specific tissue or organ. They have an innate ability to promote anti-apoptotic, pro-angiogenic, and anti-fibrotic activities, and they have immunomodulatory and anti-inflammatory effects. You can't even say too much about the fact of the immunomodulatory and the anti-inflammatory effects. And then they also have a myriad of proteins that are created in there and are also released. What they found is that umbilical lining stem cells have 2 to 3 times the capacity to clone themselves versus other cells. Well, what difference does that make? It makes a difference because if you're growing cells, you can create a higher concentration of cells with less passages. You do not have to duplicate them as much, and your cells don't get as old. Exosomes versus microvesicles, and this is probably what you're going to know the most. Microvesicles are shed from the plasma membrane, from the outer surface of the plasma membrane, and they contain components or fragments of protein sequences. Whereas an exosome originates from an early endosome, and as they mature into late endosomes, they turn into interluminal vesicles, which are multivesicular bodies. They become exosomes, and through exocytosis, they are put out of the cell. The point being, your microvesicles come from the outer surface of the plasma membrane, and they are components. An exosome comes from the endosome inside the cell that was created because the nucleus told that endosome to create a specific product. It is completed, it is activated protein, and it's completely different than just basic extracellular vesicle.